Hello, everyone. Thanks for joining the webinar today. Before we get started here with our speaker, I will say a few things and introduce our seminar crew. Starting out, our presenter, Alan McLean, who's a graduate research assistant at Michigan State University DOE Plant Research Laboratory. We have a number of panelists today to help answer questions as quickly as we possibly can as Alan speaks, starting with Aaron Sadoff, who was a key contributor to the DAT technique. He's a senior application scientist. Also, Doug Lynch, also a senior scientist in R&D here. CC Hall, an applications analyst. Brent Clausen, director of international group. Juan Michelli, international operations manager. Brandon Haney, a technical sales representative. Those are our panelists today. I'll be your moderator. My name is Rod Matson. I'm a senior product manager. And behind the scenes, we have Vanessa Wamsley, who's sort of organizing everything and make sure this production goes as smoothly as possible. So she's a senior marketing specialist here in our environmental group. And so I'm going to go ahead and introduce Alan McLean, and then I'll stop sharing my screen. Alan's going to present the following topic here. So Alan, I'll let you, I'll stop sharing, let you share your screen and take over. Thanks, everyone.
Hello, everyone. I'm Alan McLean. I'm a graduate student for Tom Sharkey at Michigan State University. And I've been part of this long distance collaboration with LI-COR, trying to test and evaluate the dynamic assimilation techniques while using it to test some hypotheses about photosynthesis in general. I'm going to walk you through some of my projects studying the triose phosphate use limitation of photosynthesis, and then I'm going to talk about what DAT added to my project and maybe how it can add to yours. Our lab is a basic photosynthesis lab, which means that we make a lot of ACI curves. It's effectively an enzymatic assay done in vivo, where we track the rate of assimilation, that's A. Laser pointer. That's A here, versus the concentration of substrate, which in this case is the internal pressures of carbon dioxide. There are three fundamental limitations to the photosynthetic rate. The first is the rubisco limitation, in which photosynthesis is limited by Michaelis-Menten kinetics with CO2 as substrate and O2 as a competitive inhibitor. You tend to experience this limitation if CO2 levels are low when testing your plant, or if the stomata are very closed. The second, this blue line here, is called the RUBP regeneration limited portion of the curve. Sometimes we call it J limitation, where the products of electron transport rate are what limit photosynthesis. That's ATP and NADPH. And those are used in the regeneration of RUBP. And you tend to experience this limitation heavily if light levels are low. The third is the triose phosphate use limitation of photosynthesis, which is where the photosynthetic rate is limited by the rate at which products of Calvin-Benson cycle can be used up. And it's best described as competition between the Calvin-Benson cycle and the electron transport rate for inorganic phosphate. You only experience this limitation when photosynthetic rate is very high, when photosynthesis is going fast relative to the normal photosynthetic rate of the plant at ambient conditions. That can be exacerbated with elevated CO2 and light, reduced oxygen concentration, or low temperature, because the enzymes that produce the end products of photosynthesis are very temperature sensitive. Here's a better explanation of what we mean by triose phosphate use. It's not just triose phosphates. Anytime phosphorylated carbon is dephosphorylated and permanently incorporated, contributes to what we call TPU capacity. I'm going to be using the acronym TPU a lot in this presentation, and that's just going to be referring to this phenomenon. So first here is the addition of CO2, creating additional phosphorylated carbon in the stroma. This next step here, the conversion of PGA to triose phosphate, involves a dephosphorylation, but since it doesn't incorporate any CO2 into end products, we would say that this doesn't contribute to TPU capacity. Everything else here in red, though, counts. Mostly it's going towards starch and sucrose production, but there's sundry other uses for phosphorylated carbon. One of the other surprisingly big contributors to TPU capacity is the cytosolic export and use of photorespiratory intermediates serine and glycine, which began as phosphorylated intermediates in photorespiration and then do not return to the plastid. there's a flux imbalance in phosphate in TPU limitation. More phosphate is being incorporated into carbon structures than is being released. It isn't just a concentration issue. A common question is, could TPU limitation be solved with better nutrition? And it can't. Firstly, the phosphate levels in the stroma are highly regulated by the vacuole. And secondly, the greater pool size wouldn't solve the flux imbalance. Now, there are a couple of interesting quirks about TPU limitation. Here's our model for what happens during TPU limitation. Essentially, the Calvin-Benson cycle is producing organic phosphates. You're competing with the ATPase for phosphate. And then because the ATPase is slowed, you end up with a limitation on photosynthesis. So the use of organic phosphates releases phosphate and then relieves the competition. Now, it's interesting, because the limitation is not on rubisco, Michaelis-Menten kinetics mean almost nothing while you're TPU limited. The photosynthetic rate becomes insensitive to CO2 or O2 changes. And that's one of the primary diagnostic things that we see during TPU limitation. So what's the point in studying TPU? TPU is the maximum rate of photosynthesis, and no matter how the inputs are changed, the photosynthetic rate can increase. As I just mentioned, you can't add more CO2 and it starts producing more. And you can't add more light and you can't take away O2 to improve production whatsoever. So it's really just a wall. So if we engineer plants to have a greater photosynthetic rate, we might just hit a wall in the future. One of the dirty little secrets is that we've probably gained some amount of productivity globally with the elevated levels of atmospheric CO2 right now, but there's no guarantee that that trend will continue. Here we have 4 different plants that have completely variable responses to elevated CO2. Potato here has elevated TPU capacity when it's grown at elevated CO2. But here, the brassica, I believe this is a cauliflower in this experiment, had overall reduced photosynthetic capacity. One of the more interesting ones, I think, is this is common bean. And if you look across, these arrows represent the concentration of CO2 at the ambient conditions at the growth CO2. If you look across here, it has almost exactly the same photosynthetic rate at the ambient CO2 between the two conditions, even though the overall photosynthesis is clearly affected. So this leads to a pretty big debate about TPU limitation and what role it plays in current and global models, current and future global models of CO2. Now, we know that plants can be adaptive in cold temperatures or low CO2, and plants have not yet been found TPU limited in the wild. Here are 2 examples of adaptation to TPU limitation. This plant, when grown in the cold, produces additional amounts of sucrose phosphate synthase, which raises its TPU capacity because it's able to produce more sugar. When it's grown partially in the cold, it produces a little bit more. And when it's grown at ambient conditions, it's relatively low. Here, plants that are placed in low CO2 reduce their sucrose phosphate synthase activity, lowering their TPU capacity to match the reduced level of photosynthesis possible at low CO2. The hashed bars here show that there's a recovery from the drop in TPU capacity, but the ability to recover from that drop diminishes over time, which shows biphasic adaptation. Uniquely, TPU limitation slows photosynthesis when carbon fixation is fast. And that leads to the question, why does TPU limitation happen at all? TPU capacity from export enzymes is very cheap compared to the amount of investment that's used for rubisco or electron transport enzymes. It must all be regulatory, which with TPU capacity regulated to match the environment, and some component of TPU limitation being important to the overall regulation of photosynthesis. So if we think that TPU is an important part of regulation, I'm going to be showing you some situations in which that regulation fails. A few years ago, when I first got interested in TPU limitation, there were a couple of papers talking about a 1-point fit for VC max. By turning off the light for a brief period of time, here it's 60 seconds, before returning the light, you can achieve an overshoot, where the rate of photosynthesis that you achieve beats the steady state photosynthetic rate. This overshoot was being used to fit VC max, the Michaelis-Menten Vmax for carboxylation. We thought, okay, probably in the dark, you're letting the dark reactions run, using up the organic phosphates and producing that last little bit of photosynthetic product before everything shuts down. Then when you return, because you freed up all of your phosphate, you can achieve a greater TPU limit on photosynthesis, because there's just some additional amount that you can achieve before you run out. So, so if you turn off the CBC, then you're producing phosphate because you're no longer producing organic phosphates. When we tested these overshooting at, we tested at 2% oxygen and 750 ppm CO2, we saw that it actually caused these dramatic oscillations above and below the steady state assimilation rates. These oscillations have long been associated with TPU limitation, so we thought we were on the right track and that the overshooting was due to essentially testing TPU limitation. But to prove it, we had to make estimations of what we thought was this higher TPU capacity. And we had to prove that it wasn't just fitting the rubisco Michaelis-Menten kinetics. We also wanted to know whether in total the plant was performing better than steady state at TPU limitation, truly exceeding its limits by perhaps cycling pools from trough to peak. So that's how we came to be interested in RACER. We wanted to make very fast measurements of the limitations to photosynthesis. So I'm just going to give you a little bit of background here on RACER. RACER was I should tell you what RACER is. RACER is rapid ACI response. RACER was the first attempt at dynamic assimilation. The scheme basically went like this. You can achieve an ACI curve very quickly by ramping CO2 over a range while taking data every few seconds. The problem was that due to the differences in path length between the LI-COR console and the two ergas, which are used to calculate assimilation, there was a blank assimilation measurement. So at the typical recommended ramp rate, this is usually plus minus 10 micromole carbon per meter squared per second. So what you would have to do is take a blank measurement of assimilation. That's, I can show you that here. Here is the blank assimilation with no leaf in the chamber. And then you would take a measurement with the leaf in the chamber. That's this black dotted line here. And you would essentially blank subtract. You would subtract this line from this line, and you would end up with an actual ACI looking curve. So the ramp rate of 100 parts per minute, parts per million per minute meant that you could finish a full ACI curve in 15 minutes or a partial in 5 minutes. Well, we guessed that the slow assimilation rate during the ramp up would be lower than the steady state assimilation during TPU limitation. It would allow for pool sizes to empty, and you would achieve something similar to what you could do by putting the plant in the dark. And that would allow for an overshoot. So then we used the RACER data to analyze the limitations to photosynthesis. Instead, we just didn't see anything. It just looked like nothing. So we put the experiments aside for 3 years. On top of not providing the answers we wanted, RACER was really difficult to use, at least without serious scripting to back you up. I know Joe Stinziano made an R script to help work these up, and I made a Visual Basic script that did the same, but I never distributed it. Without these tools, you were left to break up hundreds, if not thousands, of data points by hand. It was really, truly miserable. You dig out your forward and backwards blanks, and they had to be separate, and they had to be made each day. You decide which points you can use and which you can't. So if you made your blank, there was always going to be a portion of the curve at the ends where the data was completely unusable. And then you'd perform a fit and subtract the assimilation for the blank for your proper curves. And at the end, you got a nice looking curve. Without scripts to help you, the data analysis could take several times more time than running the curves. So now with the release of dynamic assimilation techniques, the LI-COR just does all the heavy lifting in creating these nice dynamic ACI curves. Essentially, it just abandons the assumption of steady state from the mass balance equation. It calculates what's called this accumulation term. And it prevents the need to make the blanks. It does a very good job of cleaning up the shape of step changes. Here's a step change in assimilation. The purple line here is the calculation made by the steady state assimilation, which is what you'd be used to if you aren't running dynamic assimilation techniques. Here's a step change. You see huge perturbation up to 40 micromole per meter squared per second of assimilation. That's just not existent because the plant isn't making that change. That's just calculated by the machine. And with the dynamic measurements, well, you can see the difference in scale here. The green line is the dynamic assimilation, and it looks pretty much just flat. There's a little bit of noise though. And then during the blank, here's the plus minus 10 micromole per meter squared per second that you expect from the blanks in RACER. And the dynamic just ignores it. It's just completely flat. So it actually saves you hours, maybe hours per curve for fixing your data. And it makes for just an overall superior ACI technique. There's still a little bit of pre-processing that you have to do. There's a user table configuration that you have to do pretty much just once. It's recommended that you test it maybe weekly, but I never see any changes. And then you also have to do the range match, but you had to do that before as well. The big relief is the post-processing, which was the biggest deal in terms of usability. And then the other big change is that the recommended ramp rate has been expanded. So with RACER, you were generally in questionable territory if you ramped faster than 100 parts per million per minute. With DAT, 400 parts per million per minute max is recommended, but I've used 500 parts per million per minute without issue. Just don't tell the LI-COR people that I told you that. And with the higher ramp rate, we were able to see overshooting of TPU limitation during CO2 ramps. I'm just going to separate these out so you can see that the 100 parts per million ramp behaves almost exactly like the RACER data from before, but the other curves break above the steady state ACI, just where the steady state ACI reaches TPU limitation. The peak of the DAT curve, the DAT curve, increases with the ramp rate, but so does the depth of the following trough. So this fits perfectly with how we modeled how the overshooting happens. The less time that the plant has to build up phosphorylated intermediates because the ramp is faster, the more carbon it can fix before it becomes TPU limited when it should theoretically be TPU limited. And then the assimilation rate drops. The depth of the trough increases, and this is theory, because the overall rate of photosynthesis is still limited by the overall rate of triose phosphate use. Anything that you've built up in this peak has to be exported at some point. With DAT, we can also prove that the oscillations are associated with entering TPU limitation. There are two ways that we've tested this. In the middle, we have standard ramps at a rate of 400 parts per million per minute CO2, starting from 50 ppm and working up. We've then done other ramps at the same ramp rate where the plant will not become TPU limited. On the top, we have ramps run backwards. The plant is acclimated to TPU limitation, so it doesn't enter TPU limitation during the ramp, and there are no oscillations. On the bottom, we've used elevated temperature to prevent the occurrence of TPU limitation, and that prevents any oscillations from occurring, even though the plant is ramped forwards at the same speed as it is here, where you see oscillations. So how does the dynamic data compare to steady state? Well, we plot the fit curves from the steady state equation, from the steady state ACI to show exactly what's being exceeded. We expected the plants to dynamically exceed TPU, but it was definitely a little bit surprising that we could dynamically exceed the light limited curve. And we think that it's just any available extra pool of RUBP plus any available pools of NADPH and ATP can be used to fix carbon in excess of what would normally be possible. It never gets past the rubisco limited portion of the curve, but it gets close. So if you remember, we originally kind of set out to prove that the 1-point method of estimating VC max wouldn't be correct, but I think we actually provided pretty clear data that supports it. We had to look at whether the plant was overall gaining carbon or losing carbon. Our expectation was that the overshooting was that it's carbon pool-based temporary overshoot. This wouldn't allow for any overall increase in carbon. The extra amount of carbon fixed during the overshoot would have to be worked up at the same rate as all organic phosphates. So we expect it to be at best, neutral in total carbon assimilation. We tested here two different styles of overshooting, one full ramp and a spike in CO2, both of which induce oscillations. Neither option gains any carbon over the steady state. In fact, the ramp loses a ton, 20%-ish. So you're just borrowing carbon from the future. It doesn't actually beat TPU limitation when looked at with a long eye. I think that there's such a the spike is basically no different from the steady state. It doesn't beat it and it doesn't lose to it. And there's such a difference between these two. And the reason why I think that is because there are some very fast regulatory responses to TPU limitation. And it can catch up with the spike. But when there's constant pressure being added to the system during the ramp, I just think that the regulatory components can't catch up. So if we wanted to look at these very fast regulatory responses to TPU limitation, you have to make some amount of optical measurements. This is the instrument that Dave Kramer's lab built for us to combine gas exchange with optical instruments. Essentially what we have here is a 3x3 clear top chamber with 3D printed parts of the top and the bottom of the cuvette. We have detectors built into the backplate, which is right here. And there's a little port right there for connection to a spectrophotometer. And at the top here is an LED array with a scattering optic that fits into the top of the cuvette. So we have actinic blue and red lights and green lights for measuring the electrochromic shift and photosystem 2 fluorescence, and then far red lights for the measurements of PS1. I want to briefly introduce you to the measurements that we're going to be making here. This is what we call a DIRC, meaning dark interval relaxation kinetics. We make use of the Witt effect. Basically, a pigment has a slight change in absorbance based on the charge across the pigment. So if your pigment is embedded in the thylakoid membrane, its absorbance will change in response to the buildup or the relaxation of the thylakoid delta pH. Here, we briefly put the plant in the dark to dissipate the delta pH and fit the decay of absorbance to an exponential decay. The principle of the decay is the total electrochromic shift, which is a measurement of the proton motive force. That's basically just the distance from the top here to the bottom. The initial decay, which is just the slope of this, is what we call the velocity of protons moving across the thylakoid membrane. And then the decay constant is called proton conductivity, which is GH plus here by analog to electrical conductivity. The same techniques work for PS1, but the absorbance, the LEDs that we use is different. So we can use pulses of CO2 to begin TPU limitation and then study what happens in the electron transport chain to determine what regulation is taking place and why. This gets pretty complicated. I'm just going to try and iterate on it. First, the protocol. Plants are given a pulse of CO2, which you can see right here. And then we wait for a period of time and we make our optical measurements. We do this twice. The first time we do a DIRC for the electrochromic shift, and then we make our PS2 measurements. Then we reset to ambient CO2, and we run the measurements again for PS1 measurements. Then we let the plant readjust to ambient conditions and repeat with another interval. That's how we get this time course here. We just scatter the intervals that we're waiting and then make measurements on multiple plants. By our model, the first impact of TPU limitation is the restriction of proton flow at the ATPase due to competition for inorganic phosphate. flow isn't stopped, but it's constricted. The parameter here is measured from the decay of the electrochromic shift. And after CO2 is flowed in, conductivity tanks. And that indicates restriction of the flow of protons across the ATPase. It's really clear that I say it's the restriction of the flow. Nothing is actually stopping here. It's just the rate is going to be constricted due to competition for phosphate. What we think happens next is reduction of PS1. This can only happen for two reasons. I should say, here's the reduction of PS1. We measure the PS1 oxidation state, and right at the bottom where proton conductivity is the lowest, the reduction state of PS1 is the most reduced. So only two reasons that PS1 can become oxidized. Either there are more electrons flowing onto PS1, or there's an inability to remove electrons from PS1. We measured the electron transfer rate from the cytochrome B6F complex to PS1 just to prove that it comes from the acceptor side. So this is fit from the initial decay of the PS1 signal. We can see that the rate of electrons flowing from B6F onto PS1 is slowing down. And that means that since the rate of electrons flowing onto PS1 isn't increasing, but it's becoming reduced regardless, there must be a slowdown in the rate that electrons are being taken off of PS1. So that means that there's probably a buildup of NADPH and therefore a lack of NADP. So one way that you could run out of NADP is that the reduction step happens after phosphorylation in the Calvin-Benson cycle. So if you run out of ATP, you cannot have enough flux through BPG into GAP to soak up any NADPH. So then you have buildup of NADPH and reduction in NADP. And again, these steps aren't stopped. It's just the flow through here, the flux is reduced, and that's what's causing these effects. To demonstrate this acceptor side limitation of PS1, we can look at the PS1 kinetics. So here's the protocol. To measure PS1, you measure the change in the absorbance at 820. This is the Witt effect that I was mentioning earlier. The higher the signal, the more oxidized PS1 is, and the lower the signal, the more reduced. So you have your steady state here. Then a dark period is applied to reduce PS1. This is the DIRC that we fit the decay from. Then a saturating flash is applied to fully oxidize PS1. It should drive all of the electrons off of PS1 and onto the acceptors. The decay of PS1 signal during the DIRC tells us the velocity of electrons at the PS1 and the relative position of the steady state on this scale of reduced and oxidized tells us the oxidation state of PS1. This kinetic looks pretty much exactly like what you'd expect. PS1 is reduced in the dark and then oxidizes during the saturation flash. And then it returns to a normal level of oxidation in the normal light. So this kinetic was taken from this point in the curve, where all of the major disturbances in PS1 oxidation following TPU limitation are gone. So here, this point is taken from partially up the bottom of the trough in PS1 oxidation, recovering from the most severe perturbation on PS1. And here what we see is that the signal drops during the saturation flash, indicating that PS1 is reducing again when it should be strongly oxidized. That means that electrons are building up on PS1 because they aren't being taken off quickly enough. And then here at the very bottom, where it's the most, where in the curve we saw that PS1 is the most reduced, we can see that re-reduction is very strong. There's not even close to getting rid of all the electrons that's accumulating, even during the saturation pulse. So this tells us that absolutely the PS1 is not able to get rid of all of its electrons, and there must be something happening that is preventing it from getting rid of its electrons. And we believe that that is lack of NADP. After PS1 is reduced, the rest of the electron transport chain follows suit. This is QL, which is a measurement of the oxidation state of QA, which is one of the very first electron acceptors coming from PS2. When you run out of NADP, you get reduction of PS1 that builds up back pressure, and it reduces all the way back as PS2. Nothing is being done to increase the flow of electrons from photosystem 2. The light hasn't increased, and instead it has to be the inability to pass on its electrons that causes reduction of QA. QA redox state acts like a valve. It has to be open for photochemistry to continue. And if it's closed, all of the energy is instead shunted into non-photochemical quenching. Here, with the reduction of QA, measured here as QL, we see a huge drop in phi2, which is the ratio of light energy used for photochemistry. Conversely, there's a huge increase in the yield of non-photochemical quenching. And all of this is because the valve, which is QA, is closed. Some mechanism has to eventually stop these oscillations. And I believe here it's the proton motive force dependent non-photochemical quenching. PMF, which here is measured as the total electrochromic shift, increases monotonically, and so does NPQ. So here is ECST, which is a measurement of PMF and non-photochemical quenching. So more energy is being driven into NPQ by slower to adjust processes, which slows the effect of the fast responses. The other component here is photoprotection from cytochrome B6F, which here is KET. KET is the rate of electron flow from B6F onto PS1, and it slows down at higher PMF. So these two PMF-dependent forces slow down photosynthesis and quench these oscillations over time. So in conclusion, high ramp rates or spikes in CO2 allow previously empty pools to fill quickly, allowing the plant to dynamically exceed the limitations on photosynthesis typical of the steady state. When these pools fill quickly, phosphate concentration drops dramatically and causes constriction of the electron transport chain, accompanied by reduction of PS1 associated with NADP depletion. And then over time, these oscillations in electron transport and assimilation are damped by PMF-associated non-photochemical quenching and photoregulation at cytochrome B6F. So now I'd like to talk about something completely different. ACI fitting models are over-parameterized. You don't have to actually know any of these equations, although maybe you should at some point if you do a lot of ACI curves. Here are all the parameters that we use for fitting ACI curves. There are a couple of ways you can consider this. It's one data set fit with one model with 7 parameters, or it's one data set fit with 3 models in a step function, each with up to 5 parameters. So if you have a typical ACI curve, you probably have 16 data points or 18 data points, or maybe 20 if you're stretching it. And if it's one data set one data set of 16 data points fit with 7 parameters, that's quite a lot. Here's a pretty normal ACI curve on Benthamiana at saturating light, which is 1000 micromole per meter squared per second. If you consider the fitting function as one function, it's 16 data points with 7 parameters. If you consider this a step function, then if you look at the RUBP regeneration limit portion of the curve, you have 4 data points being fit with 5 parameters. Mathematically and intuitively, this feels very flawed, and you'll need additional data input to achieve a good fit. Some things that you can do are, you can do another curve at lower light levels, which will allow you to fit mesophyll conductance a lot better. Other things like that, you can do C14 analysis to determine mesophyll conductance before you begin your ACI curves. But the point is that it needs additional data input. Now, ACI curves are fit non-linearly. The algorithms can't just find one global minimum. And instead, usually what they find is the closest local minimum. Now, overparameterization is only strictly defined for certain polynomials, which the ACI curve model just isn't. The ACI curve model really isn't even a function because one of the parameters affects the x-axis variable. And it's also a step function with floating breakpoints. So rigorous statistical analysis really isn't an option. However, there's one way that we can assess overparameterization. It's possible to fit the curve. Is it possible to fit the curve with multiple starting conditions for the fit and end up with many plausible fits? Here are 4 fits that I want you to look at. And this is all fits to the same curve. None of the data is any different. And all we've done to get these 4 different curves is alter the starting condition for the fit. And fit 1, okay, looks pretty nice. Sum of squares residuals is 0.46. Looks like a great fit, right? BC max of 58, GM of almost 2. Looks pretty good. Here's fit number 2. Only thing that's changed is the starting conditions for the fit. Sum of squares residual is 0.432. Also a very good fit. VC max of 82 compared to 58. Mesophyll conductance is about half. Fit number 3. Sum of squares residuals 0.312. Looks like a very good fit. VC max is even lower than fit number 1. Mesophyll conductance is 3 times as high, or 6 times as high as fit number 2. Fit number 4, sum of squares residual of 1.47. Still quite a good fit. VC max is the lowest of them all, and your mesophyll conductance is 14.7. So 14 times as high as fit number 2 here. So the moral of the story is you can fit your ACI curves many different ways and come up with extremely plausible looking fits where just none of the data lines up between the fits. Or sorry, none of the fitting parameters, none of the results line up. And that's the surest sign that the ACI curve is overparameterized, especially when it comes to mesophyll conductance, by far the most variable of any of these parameters. And alongside the variability of mesophyll conductance is VC max, because in the rubisco limited portion of the curve, they can compensate for each other. So how do we stop this? I would, if you are insistent on using steady state ACI curves, you have to measure mesophyll conductance separately. You can run a curve at lower light levels, which will push many points into the RUBP regeneration limited portion of the curve. And the shape of the RUBP regeneration limited portion of the curve is the most fixed, which allows you to measure mesophyll conductance a lot better. The other option is collect a lot more data points. And that's one of the best things about DAT, in my opinion. If you run a DAT curve, if you run it as quickly as you can, which is 500 ppm per minute, and you're going from 0 to 1500, you're collecting 3 minutes worth of data points at a data point every 2 or 3 seconds. So that's 60 or 90 data points. So already you're producing 4 to 6 times as many data points. And if you run your DAT curves slowly enough that you are taking the same amount of time as you would normally run a typical steady state ACI at, you're going to be producing hundreds of data points. And that kind of data caused this one ACI curve to only have 1 global minima. So does this prevent the ACI curve from being overparameterized? I can't really say yes or no to that, but I can say that it helps. And that's all I have to say. I'd like to say thank you to all the people of the Sharkey Lab the Kramer lab, who helped build that instrument that I showed you before, especially Jeff and Robert. Robert was our electrician and Jeff was the designer. And I'd like to say thank you to the people from LI-COR. Doug and Aaron were my go-tos during our collaboration, and Rod and Mark put everything together for me and brought me here to do this seminar. Thank you, and we have plenty of time for questions.
Okay, thanks a lot, Alan. That was a very interesting talk. And we actually do have a few questions that came in, and a couple of them we definitely would like you to answer. Early on in the presentation, there was a question, do people assume J is constant during RUBP regen limitation?
Yes, that is the fundamental assumption of the RUBP regeneration limited portion of the curve. The shape of the curve comes from, let me bring up the, yes, here. The RUBP regeneration limited portion of the curve has this distinct shape, and that comes from how electron transport products are partitioned between photochemistry and photorespiration as the concentration of CO2 increases. When the concentration of CO2 is very low, most of the products are going towards photorespiration. And then as it increases, it sharply increases the amount the proportion of reactions at rubisco that are going towards photochemistry. So the assumption is that it's constant. Now, what isn't true is that J is constant between light levels. In older variations of the fitting model, what was measured was what was called J max, which was the maximum J during at saturating light levels. But we've found that that's not an accurate measurement, so we just call it J now.
Okay, and a follow-up to that, also from the same individual. How were the TPU oscillations observed during DAT?
How are they observed? So when they begin, we are just taking measurements along every 2 seconds, no matter what. So all that is happening in these curves is we are taking data points every 2 seconds, and we are ramping the CO2 at this ramp rate. And then anything that the plant does just comes out. And so the appearance of these am I doing? Do you see this?
Yeah, we definitely see that. I'm not sure what happened. There it
Beats
goes.
me. Yeah, so anything that the plant does just comes out in the data, and there's nothing that we're doing to force these, especially. Now we are using, this is done with bentamiana, and these oscillations happen very regularly with bentamiana and with tomato and tobacum. All these solanaceae, which have a very brief period between the RUBP regeneration limited portion of the curve and the TPU limited portion of the curve, all of those plants exhibit these oscillations very readily. Whereas plants like potato don't show these oscillations almost at all. Almost at all. I'm losing my voice, sorry.
Okay. Another question. They're asking, is it only from the trend of the ACI that we can say that this plant has a TPU limitation or not?
There are a couple of diagnostic features that we use for detecting TPU limitation. Let's see. The first is just from the ACI curve, just from the shape of the curve here, where the assimilation rate becomes flat or drops off a little bit. The dropping off is very diagnostic. In some cases, you can get an RUBP regeneration of a portion of the curve that looks very flat, depending on the shape of the curve. But the other diagnostic features are all done with fluorescence. So if you see a decline in phi2 with an increase in with an increase in CC, then what you have is TPU limitation. And the other diagnostic features are decreasing proton conductivity or increasing PMF. All of these features we use to diagnose TPU limitation. Probably the best one for people out there is you can look at the shape of the curve, or you can look at decline in phi2.
Okay, and a couple more questions. Can we detect different TPU limitations at different times of the day? For example, early in the morning versus the afternoon?
I can't answer that. I don't know. I have done studies where you leave the plant at TPU limited conditions overnight, or sorry, for an entire day plus a night, and then the next day as well. And you can see a decline in TPU limitation over the course of the day. But it restarts as normal at the beginning of the next day. And I can't answer whether that decline that we saw was due to the time of day, or whether it was due to the fact that the plant had been TPU limited up until that part of the day.
Okay. Here's another one. Anytime this individual has seen a reduction in TPU, they also see a downregulation in BC max and J max. In the long term, that can drive to a more permanent downregulation of photosynthetic capacity of the leaf. Can we associate TPU limitation to the strength of the sink demand?
Not strictly. There's never been a causal link proven, but that is the tenor of the discussion right now, is that it is affected. And some of the papers recently have been like the favor papers, where they reduce or they prune sink tissues, and then they see a total reduction in photosynthesis on everything. And there are old papers, like I remember King from 1952. If you remove the panicle from wheat, you end up with overall total photosynthetic reduction. And Christine Foyer's lab has been studying this extensively, or at least they used to back in the day. And they also saw the sink tissue removal affects leaf level photosynthesis across the plant. So there's a lot of experiments and a lot of data piling up that there is reduction of TPU capacity when sink tissues are removed, but it's accompanied by reduction of everything else. And then on top of reduction of everything else, we can't prove that the reduction of everything else is due to reduction in TPU. And we can't prove through some mechanistic way that reduction in sink tissue causes a reduction of TPU capacity. It just hasn't been proven yet. But if you make that conclusion in your paper, people might support you.
Okay, thanks, Alan.
That's not a very good answer, but that's the best I've got.
It's the best anybody here probably has, so we appreciate it. Another one, are you aware if there are any differences in TPU limitation with plants that are more drought tolerant or drought resistant?
I don't know. Yeah, sorry, I just don't know. I know that there have been some papers in the past where they study the effects on TPU capacity when plants become drought stressed, but I don't remember what they saw even. Sorry.
Oh, that's fine. When you compared traditional ACI curves with the dynamic ones, did you find by any chance overestimation of VC max in the dynamic curves?
In my dynamic curves, I saw, if anything, a very slight underestimation. And the amount that it underestimated was proportional to the ramp rate. So if you look at these curves, here's your steady state, which is this purple line here. And then all of the curves are ordered by speed going up here. So you can see that they fit to just a slightly lower VC max. And I don't believe that that's an instrument error. I entirely believe that that's something that's happening in the biology.
Okay. Yeah, sorry, we keep hitting you with questions, but I think you're the only one that can answer some of these. Here's another one.
This is the first time that I've done a seminar online where people seem to have paid attention.
Yeah, and there's definitely a lot of interest here. Here's another one. When you run these DAT curves with the intent to fit the curves, do you recommend starting at low to high, acclimated at 400, or high to low, acclimated at around 1500 to dampen the TPU oscillation?
Yeah, this is where we get into tricky territory. If you want to avoid oscillations, I believe that the best practice is to go low to high, but do your ramp very slowly. If you go very slowly, you'll get tons of data points, which is just a fun little bonus, but you can avoid the oscillations. You can, and I recommend that you try ramping it a bit quicker, depending on your plants. If it's solanaceate, you have my sympathies, because you're probably going to get oscillations like this. And I believe that friends of the lab have seen these oscillations with many other plants, poplar, I think, for instance. You can do your curves high to low, and I think that that is totally legitimate. As long as you do all of your curves like that and you're comparing apples to apples, it's fine. But when you start out with your plant adapted to TPU limitation, you have reduction in rubisco activation state, and you have reduction in, or you have buildup of QE. That's just going to happen. And the question is whether ramping backwards will give it enough time to overcome those before it enters like rubisco limitation, where the rubisco activation state will make a huge difference. It's something that you really have to test for yourself. I'm sorry. I really like the slow ramp forward. That's probably my number one.
Yeah, and a similar question here. The last one that's in the Q&A. In our experience with DAT, 150 ppm ramp is most of the time suitable compared to the steady state curve. I don't know if you can comment on that.
Yeah, sounds good. With these plants, the 100 ppm, 150 would also be too high, because you do get this drop here at the bottom. Let me just show you like right here. So the shape of the curve is distinctly different. And that's just because this is Benthamiana, and it's super susceptible to TPU limitation. There's a reason why we use this plant for these experiments, because it really hits them hard with TPU limitation. 150, depending on what plant you're working with, certainly no problem.
Very good. Well, that's all the questions we have, Alan. For everybody that's attending, or everybody that registered and has attended, we will actually send out an email with the recording, and also we'll also have a link to a paper that has been published on, not on Alan's content yet, but on the DAT methodology. Before we exit, I guess one more just popped up, Alan. We're at the top of the hour here, so we'll take this last one. How does DAT do with water and CI estimates?
So this is actually a pretty interesting question. Generally speaking, if you're doing a DAT curve, the water isn't changing fast enough for it to be a problem. Now, the dynamic equations are prepared to handle dynamic changes in water. It's just a question of whether you should be using the dynamic measurement of water or not. I would probably just use the steady state measurement of water. It's certainly fast enough. And then if your dynamic calibration isn't perfect, it doesn't make a difference. But if you use the dynamic measurement of water, no problem as well. Just make sure that the calibration works out.
All right. Thanks so much, Alan. Thanks everybody that attended today and all the panelists. And again, we'll send out a recording. If there are any additional questions that come up as you think about things, feel free to reach out to the LI-COR. And we obviously can get a hold of Alan and he can work on any of those questions to clear things up.
Yeah, you can contact me at mcclai54@msu.edu. Please send me any questions that you have.
All right. Well, between LI-COR and Alan, we'll get any other questions answered as we move forward with this methodology in the instrument. Again, thanks everyone for attending. Thanks again, Alan. Everybody have a great day.